Assalamualaikum and selamat sejahtera. Today we continue our lecture Inheritance and Genetic Control in the Non-Mendelian Inheritance Part 1. And in this Part 1, we will discuss about codominance, incomplete dominance, multiple alleles, polygene and ladder alleles. Next, for the Part 2, we will discuss about linked gene, sex-linked gene and also the genetic mapping. Non-Mendelian means deviation from the Mendelian inheritance. Inheritance of character by a single gene may deviate from simple Mendelian pattern in the following situation. First, alleles are not completely dominant and recessive. Second, a gene has more than two alleles. Third, gene produce multiple phenotypes. Fourth, trait may be determined by two or more genes, or we call it as polygene. Fifth, leader genes. And another factor that will not be discussed in this topic is linked genes, sexual link, also the link by the chromosomal link of the genes also will contribute the deviation for the Mendelian inheritance. What is meant by deviation from the Mendelian inheritance? Mendel give when we make cross of the homozygous, monohybrid and dihybrid of the heterozygous, we will get for the monohybrid we will get the ratio is 3:1 ratio for the F2 generation. But in this deviation, we will not get numbers. We will not get the numbers, and also for the hybrid also, we won't get the 9:3 to 3:1, and we call it as the deviation of the Mendels. First deviation of the Mendel is codominant alleles. Codominant means both alleles of a pair are fully expressed in front of heterozygous. It occurs when both of the contribution of alleles are visible in the phenotype. That means the both allele there is no recessive. Okay, both character will be shown in the phenotype. For example, in MN blood group and also ABO blood group. We discuss about MN blood group or blood type. The blood type of MN is about the presence of protein in the cell membrane of the blood. If there is M-type protein, that means if we react with anti-serum M, and the blood will clot. So in this case of M group, the genotype is homozygous LM. We write like this. Like this, LM. And if the blood in the heterozygous MN, so we write like this, LM and LN, and so on for the N, LN. So what we are going to focus is here, MN blood group. So if one of either M and N is recessive, it should show one character only. But in this case, both R react with the antiserum M and antiserum N. So actually, these both are shown the phenotype, their phenotype. So this one we call is both dominant or co-dominant. Okay, let's try to cross between MN and MN group. So what we have here is the phenotypic ratio is 1M group, 2 MN group, and 1 N group. So ratio is 1 to 2 to 1. We ask Mendel 3:1. So it shows that this one is deviate from the Mendel. So it's called the non-Mendelian. In the ABO blood group, the codominant is shown by A and B. A and B type phenotype is actually codominant, and O is recessive. Because when O is by itself, of course, it will show O, but when O with A, it will show A blood type. That means O is recessive. But in the presence of B in A, it won't show the A or B. It show AB. That means both are dominant. Okay, this is because A and B will produce carbohydrate at the membrane of the blood. So for B group also produce a carbohydrate B, and A group will produce carbohydrate A. So this one show the codominant. Incomplete dominant alleles. A form of intermediate inheritance in which one allele for a specific trait is not completely dominant over the other alleles. Example in the snapdragon flowers. So the difference between the codominant and incomplete. The codominant will show both characteristic for the both alleles, but this one it show the intermediate. For example, this flower. Let's say the dominant is R, capital R is red, recessive is small r white. But it exists in the heterozygous. When it exists in the heterozygous, this flower do not show the color of dominant, that is red, but it show the intermediate between red and white, that is pink. So that one we call is incomplete dominant. So where we are going to make to write a cross between these incomplete dominant, we can write like this. We put the C and R. R is is the allele. Okay, CR. This one is, that means is dominant. And for the recessive, let's say here white. Okay, CW and CW. So in this cross here, we can see here we get the firstly we get the offspring of F1 also is not follow the Mendel's because it's not show the dominant characteristic of the flower when it exists in the heterozygous. For the Mendel, of course, it will show the dominant when it exists in the heterozygous. And one more thing, when the F1 generation is crossed, that means the monohybrid cross, this also do not produce 3:1 ratio of the phenotypic ratio. It's produced 1:2 and 2:1. That means one red, two pink, and one white. And this also proves that this one is not follow the Mendel's, or we call it as non-Mendelian also. Another cause of the non-Mendelian is multiple alleles. One gene having more than two alleles, all alleles control the same characters. Example, human ABO blood group. That means two alleles is normal. If we use Mendel, normally we have recessive and dominant, two alleles. But in this case of multiple alleles, there might be three alleles that control the same characters. For example, the ABO blood is controlled by A allele, B allele, O allele, and then we will get four types of blood group. For the blood group of ABO, it's controlled by three allele, A allele, B allele, and O allele. Right, like this, IA, IB, and I. The difference between of the blood group is the presence of the carbohydrate on the cell membrane of the cell blood cell. which is if the blood A is will contain the carbohydrate type A, and also B contain the carbohydrate of type B. So the genotype for the blood A might be is come from the uh, IA both, or IA with the O group, or IAI. That means here IA is actually is recessive, and also for IB and IBII also give the group B. But when the presence of the allele A and allele B in the genotype, it will be shows both characteristic, that is A and B. So for the O blood, actually there is no carbohydrate, okay, no carbohydrate around the cell membrane, and we named it as O. So here we have from 3 alleles, 3 genes, we have 4 groups of the blood. This table show how to test the blood group. For O blood, there is anti-A and anti-B. That means there is antibody that will react with carbohydrate type A and carbohydrate type B. Of course, in all blood A, B, and AB, there is the type of carbohydrate. That means this O blood we clot with all of 3 type of the blood except itself. And that means O only can accept O, and they cannot accept A, B, and AB. And so on. A and B blood group also, they will react the same thing, where A have antibody B, and it will be react with any presence of carbohydrate carbohydrate B, either in B group or in AB group. But for the AB blood, there is no antibody. That means it can accept any blood. Okay? Any blood, it when it's mixed together, it won't be clogged. So here we can say that O is a universal donor, and B is a universal acceptor. So O can give to everybody, to A, to B, to AB. Let's try to cross between two parents of the monohybrid, which one is have heterozygous B group and another one is heterozygous A group. Okay, look at the allele here, B allele and O allele. Okay, and also for the mother, A allele and O allele. And then we cross between these two gametes, sperm and eggs. So we will have offspring. One is will show that AB group, one will show the B group, one will show with A group, and last one is O group. So we have the phenotype ratio here is 1, 1, 1, and 1. So this show that these multiple alleles of the ABO group also deviate from the Mendel's. Okay, let us compare between this complete dominance of one alleles, incomplete dominance of either alleles, codominant and multiple alleles. So for the complete dominance, heterozygous phenotype same as the homozygous phenotype. It's like look like here, when this homozygous capital P and heterozygous capital P and small p, it really show purple here. That means it has same phenotype both. And then for the incomplete dominant, heterozygous phenotype intermediate between the two homozygous and phenotypes. Okay, so this one is the dominant, recessive, and the heterozygous is intermediate at the middle, either this characteristic and characteristic, and half of come from this characteristic. But for the codominant, both phenotypes express it's heterozygous. For example, in the ABO blood groups, where IA and IB give the carbohydrate in the cell membrane, and you give the both will react with the anti-A and anti-B. So it shows two types together. For the multiple alleles, in the whole population, some genes have more than two alleles. So this one we still not yet discuss about the multiple alleles. Yes, we already discussed for the multiple alleles. Whereas we also can give the example of the blood group IA, IB, and I, because here we can have three alleles for one type of the character. Compared to another, we have only two. Okay, two alleles for one character. Next cause, the non-mendelian, is caused by the polygenes or polygenic inheritance. Polygenic inheritance, some phenotype determined by additive effect of two or more genes on a single character. Traits controlled by two or more genes are said to be polygenic traits. Polygenic means many genes. Polygenic traits often show a wide range of phenotypes. Example, the variety of skin color in humans come about partly because more than 4 different genes probably control these traits. The gene might be located in different chromosomes or in different locus. So if in one chromosome, it might be they located in different locus, which these all genes will control same traits. The polygenic trait, for example, height and skin color, are controlled by more than one gene. Instead, several genes contribute the final phenotypes of the given traits. For example, the skin color. So many genes will control the color of the skin, the pigment, the color, and lastly, finally, they all will contribute the trait of the skin. All genes will contribute a small amount to the phenotypic that shown by this organism. Each gene plays a small role in the expression of the trait. This allows for the trait to vary greatly from person to person. Height is polygenic trait, which is why people can be very short, very tall, or anything in between, like this. Polygenic characteristic is a result of cumulative effect of few interacting genes. Then many of these discrete phenotypes can be combined together to form continuous variation. So the continuous variation is the graph with a normal distribution like this, which we have extreme, both extreme, side by side, and the intermediate characteristic at the middle. So we can say that polygenic characteristic is often quantitative. That means the characteristic is easily being affected by environmental factor. For example, the diet, the quantity of light, and so on. Normally, the allele of the quantitative polygenic will have the dominant and also recessive. For example, the dominant is for the tall in the trait of height, and the recessive is for the short. So because of so many allele of the characteristic involved in these traits, for example, A allele, B allele, and C allele, altogether will contribute the height of a person. So if all the allele is dominant, so the person might be very tall. And let's say if the person, one of the allele is recessive, only one, so it will be shorter, and so on. is many more recessive, the person will become shorter and shorter and shorter and the shortest one. So this contribution of this allele phenotype will give the trait of a person, either tall or short. As the conclusion, the resultant phenotypes of the polygenic characteristic is usually the product from the interaction of genotype and the environment, where the genotype determine the potential of organism, whereas environmental factor will expose and determine either the potential can be achieved or not. For example, if they have the gene for the tall, but if the person have a bad diet, they cannot be tall. But the environmental factors, if we have good diet for tall, and then the person become tall. Leader genes. There are several conditions where the phenotypic manifestation of certain genes could lead to death. Such genes are called leader genes, which occasionally arise from normal gene by mutation. Genes which result in the reduction of viability of individuals or become a cause for the death of individuals carrying them are called as leader genes. Certain genes are absolutely essential for survival. Mutation in this gene create leader allele. Example of the leader gene can be shown in this cross of mice, where they use the yellow mice which has the heterozygous genotype, which is one dominant and one recessive. Normally, this cross will produce 3:1 ratio, where 3 will show the dominant phenotype and 1 will show the recessive phenotype. There is yellow and non-yellow. But in this cross, they only produce 2 yellow and 1 non-yellow. And this is, of course, not follow the Mendelians. Actually, where is another one? Then they make a dissection of the uterus of the mice during the pregnancy. They found that another color in the uterus is that is red color. And then firstly they made a conclusion that actually the red is the dominant color, not the yellow. Yellow is actually the intermediate color between red and non-yellow. So this color allele is actually incomplete dominance. But why the red color is dead? This is because in the red allele, red color allele, or in the dominant allele color of color, there is another genes in the allele. There is the leader genes. So when the leader gene exists together as monozygous in the dominant, it will show their properties, and the offspring die in the uterus before it's be born. Then in the recessive of color, capital small y, or in the non-yellow, there is a survival gene that is dominant to the leader genes. So when it exists in heterozygous, the survival gene will mask the leader gene and the mice will survive. Another example that of the leader gene that happen in human population is Huntington disease. What is Huntington disease? Huntington disease, or sometimes called as Huntington's chorea, is a rare and fatal inherited disease of the central nervous system. The disease is caused by the single dominant allele, which means that heterozygous individual will develop the disease. The disease causes damage to brain cells, leading to the gradual loss of coordination, decline in mental ability, and change in personality. Because of this gene is gradually increase, show their phenotype, the person who carry this gene might be quite enough time to get married and give birth a baby, and it can be inherited from generation to another generation. Normally, the people who have this kind of gene will show their disease at age of about 40, like that, and of course, some of them already have babies. For example, in UK, around 4,800 people have Huntington disease. When a person uh, affected by this disease, they carry the gene in the kind of dominant genotype. So this genotype not necessarily for the Huntington's strictly. Maybe this genotype is for skin color or whatever. But there is gene inside this dominant phenotype. When this person marries to a normal parents, this disease is not shown for the early age, and then when they can give this gene to his affected to his son or children, and because it's the allele of dominant, so in this cross we can get probability of two of their children get affected. So this disease always inherited from generation to generation because people do not realize they have this kind of disease. Suddenly when they go to the age about 40, the person will get sick and they will lose coordination, sometimes about nervous system problem, and lastly they can detect that they have Huntington disease. And that time maybe it's already too late because they already have children like that. So I think that's all for this part one lecture of the non-Mendelian. And I know it's very difficult actually to understand when we discuss like this, and we still have time to discuss it back when we go to school again. And you can ask me in WhatsApp or just call me any part of this lecture that you do not understand, because it's quite new and some of the explanation is probably quite confused, because we have no two-way interaction. So please, you can have more understanding in your textbook here, and they have a very good explanation, and please read it frequently and make sure you understand the content of this textbook, because I found that inside this one is very complete explanation for you to understand this genetic inheritance, and I know it's very new for you. Okay, see you again. Thank you.